Everything's going.
Just give up like a 2-minute buffer. Is that?
It's on now.
No, that was all.
Everybody.
Well, I didn't. Like I'm impressing. I mean, I guess I know where the judges are. I don't think that finishing writing unless it was like
See you too, Rick. How you been?
I mean, I have this.
We probably have to running off to do something.
I didn't stay. I felt badly, but I'm going to connect with them in New York. We had this really wonderful to link everything. AI and art. I just love it. The other thing they're all like, maybe I'll do this, maybe I'll do that. I want to network. I learned how to do this. Maggie showed me how to get the QR code to link to. And one of them is numbered. I was like, okay, we'll get together, we'll do this, we'll do that, call me. Even if it's just to say, gee, what if I try this, what if I try that? So these are things I would never do. I really love
Oh yeah.
You're wrong.
Oh yeah, no, this is. I feel like
I guess it's still right down the road.
Good morning, everyone. I think we're going to start the process. Good morning. Good morning, everyone. Can we, there we go. Everyone, good morning, everyone. We're going to start our presentations this morning. I just want to introduce myself. My name is Frank Milone. I'm one of the founding partners of FML. We're a sponsor of the event. And we are a regional accounting firm, audit, advisory, tax. We have locations throughout Connecticut, and we have a location here in New Haven at District. I lead the firm's emerging companies practice, which means I lead the efforts to help startups, entrepreneurs, found companies, grow companies, scale companies, and exit companies. And it's a pleasure to be here. I'm also a Yale EIR, and I'm just here to introduce to you 27 amazing biotech companies so that you can learn more. And then if you want to get more information after their presentation, there is no Q&A here today, but you can meet with them after the Biotech Pitch Fest is over, and they'll all be here. So without any further ado, I'm going to introduce the first company, and that's Arjun Ganesan from Ixera.
Morning. You guys hear me okay? No?
Not really.
Not really. Is that any better?
Yes.
Okay. Wonderful. Yeah. So the American food producer, just by harnessing technology, they've become a third more productive than any other food production supply in the world, right? And the consequence to you and me as consumers is we're actually able to go and buy chicken for under $5. That's not to be taken lightly. But what's crazy is this last quarter, right now, the one that we're just exited, 9 out of 10 poultry companies in the United States lost anywhere between $1 million and $13 million every single week. And that is just not sustainable. So what we're doing here at Ixera is really helping companies find new margins in places that they've not found before. Right? So let's talk poultry very specifically. 3 big areas. Number 1, macroeconomic risks, war. What does that mean to their supply of corn and soybeans? Right? Number 2 is when you're thinking about the production decisions that they make. What is in their nutritional mix? How are they doing their animal husbandry? There isn't a simple, easy way of attributing things back over there. The third one is actually looking at what is in their microbial composition in their food supply, and how exactly is that impacting their productivity. So our whole thesis is if we can provide better visibility into the supply chain, we can help these companies usher us into the future, right? Okay, so some of the 2 most advanced industries, at least in our opinion, are biotech and pharma, and the other one is healthcare. And the one thing that they've done incredibly well is look at real-world data to really decide which of these decisions need to be made and which of these are just one-off data points, right? What we're doing is we've harnessed that. In the last few months of being commercial, we've monitored over 50 million chicken. So we've monitored close to about 50,000 different feces. And we've attributed that back to every single vaccine and therapeutic product that's currently available in the market. And more importantly, what we've also done is we pulled information about how these chicken are living in each of these houses, and we brought all of that back together. And the way all of this is brought in is what we call as our command and control center. So we've harnessed the information from about 6,700 USDA regulated facilities which produce food. And we've attributed about 250 data sources. And that's before we put an ounce of data into any of these locations. So we know more about the food supply chain before we walk in with the company. So the question is, where does all the biotech come in? Well, this is the framework that Ensera builds our ontology on, right? Homegrown, started at YEI a little over a decade ago at this point in time. We started the Piper. It is a industrial scale imaging flow cytometer that solves for Brownian movement. We're able to look at things like feces with molecular specificity, and we have the fastest assay in the market. So it crams down any sort of data generation down from 3 days or more down to a few hours. Number two is we've harnessed the power of sequencing, but we've taken narrow use cases, like looking at the CRISPR loci of the salmonella genome, which is incredibly well preserved, to help these companies actually make decisions. And the third one is what we're doing is we're looking at traditional diagnostics, but we're speeding it up with tremendous automation. What we're bringing is we're bringing all of this together, packaging it with our data models, and we're making it available into bite-sized information to the different personas in the food supply chain. Let's talk some examples. So we've got 3 products in the market right now. The first one is coccidia monitoring. So chicken, if you think about it, is really the business of changing corn and soybean into protein. It's a little bit like a car factory in reverse. Imagine if a car factory had the door sizes be about 1 foot apart. That's just not acceptable. Similarly, it's not acceptable to have a chicken that's between 4 and 5 pounds. Today, companies have no way of finding out until they actually see it. We're the first company that's actually helping these companies figure out 6 or 10 weeks before they actually see it in production, the impact that these microbes have on their output. And we do that by analyzing their feces and bringing the whole platform together. Our second product is around salmonella. The United States already produces some of the safest food, given the scale of food that we produce, right? But we as consumers, we have an expectation of exceptionalism. We want fewer ingredients in our food. At the same time, we want safer food. At the same time, we want these companies to be there around to continue to feed us. What we do is we provide granular information to help these companies figure out what they can take out and what they need to put in to build the future of a secure supply chain. And lastly, irrespective of whether you're a pharmaceutical company that's supplying products, or if you're a chemical company, or you're one of these large corporations, today your cadence is approximately every few weeks. We're here to bring that down within a few hours. So the whole point is, as we think of the future, we look at the next 50 years, and we have a slightly different narrative. Our whole thesis is the people that are producing food today are the ones that are going to be producing food for the next 50 years. We don't think it's just picks and shovels that they need. We think it's a whole new industry. We're here for it, and we're just getting started.
Thank you very much. Presenting next is Tim Siegert from Alex Therapeutics.
This is great. Rapid fire pitches. And early while everyone's caffeinated. Oh, all right. Sounds great. Excellent. Well, thank you everyone for coming. Glad to give everyone an update on Alex Therapeutics and what we've been up to and what our goal is. And really, we're founded and spun out of Yale, and our focus is on treating neurodegenerative diseases, specifically by targeting the synapse. So this is our company at a glance. We're a small team. Patrick's in the audience as well. Again, Steve Strittmatter, who is the chair of neuroscience at Yale, is our scientific founder. Steve Block is our CEO. I'm our president and CEO. I was a Yale Blavatnik Fellow before I joined Alex. And then Paul Fontaine is a former CEO of BI and is on our board and is really involved. We're a Series A stage company. We have a great team of Connecticut and Yale-related investors. Many are in the audience, so Connecticut Innovations, Spring Mountain, Elm Street Ventures, but we also have Bristol Myers Squibb, which is where we in-licensed our asset from. And not only do we have equity funds, but we've also synergized with non-dilutive funds, about 2 to 1, with a great track record of grants from the NIH through SBIRs, R01s, the Alzheimer's Association, and the Michael J. Fox Foundation. We've got a great board of directors, many are in the audience today, and a great SAB, including Richard Hargreaves from BMS, who gives us a lot of insight on the big pharma approach to neurodegenerative diseases. So when you think of neurodegenerative diseases, probably the one that stands out most is Alzheimer's disease. There's still about 6.5 million patients in the U.S. that have Alzheimer's disease. And though there's been a lot of news lately about lecanemab and its upcoming potential approval next month, it's still a huge unmet medical need. That drug is going to cost about $26,500 a year for treatment, and it only slows cognitive decline by about 27%. So what we're doing is we're going beyond that. We want to target beyond A-beta removal therapies to really target the underlying cause of synapse dysfunction, which are the toxic A-beta oligomers. Now, lecanemab targets that a little bit better than some of the others, but doesn't fully address that toxic pathway. And the key protein at that synapse is mGlu5, which really mediates that synapse toxicity process. And there's also a lot of overlap with other neurodegenerative diseases here. So beyond Alzheimer's, we're thinking Parkinson's disease or orphan Creutzfeldt-Jakob's disease. So our drug that we're developing, BMS 984-923, it's a little bit of a mouthful. It's a first-in-class oral small molecule therapy that uniquely targets mGlu5 in a specific way, that targets only the neurotoxic function of the receptor and preserves the normal function that's responsible for cognition. Our drug is in phase 1b clinical trials now. We've launched a Parkinson's disease program, and we're also exploring other neurodegenerative diseases like the orphan CJD. So our approach really is very different from A-beta removal. Our focus is really on preserving and protecting synapses from ongoing damage in the disease. There's a lot of clinical evidence that this target is necessary for disease progression. So mGlu5, our target, is linked to two of the genetic risk factors that are synaptic, that are known for both Alzheimer's and Parkinson's disease. And it's coupled to the other pathology in Alzheimer's, which is tau pathology, downstream of A-beta. We have a tremendous amount of preclinical evidence from both genetic and pharmacological blockade of mGlu5 signaling. So you can target this receptor and you can actually rescue animals so they're in a disease state, and then they're fully rescued back to wild type. They still have plaques in their brain, but they're fully functional in their learning and memory. And that's because what we're able to do is restore the synaptic connections between the neurons to preserve that memory. The drug itself is validated in multiple preclinical species. It's highly potent towards the target. It reduces all these downstream biological hallmarks of Alzheimer's disease, including tau pathology and synapse loss. And it has an attractive clinical safety and PK profile. So it's orally bioavailable. In our phase 1A study, we've been able to actually measure the target engagement in the brain, so it gets to the receptor in the brain efficiently. We see linear dose-dependent PK. It's very well tolerated in humans with no safety signals to date, and we've got great safety margins to the preclinical species. So a little bit about the history of ALEX and where we are now and where we came from. Maybe focusing less on spinning out in 2019, but more recently, over the past few years, in 2022, we became a clinical stage company. We launched our phase 1A single ascending dose study. We've confirmed that the drug is getting to the target in the brain. This year, we're focused on advancing and expanding this program. So completing our 10-day repeat dose studies, which are in the clinic now, and launching a Parkinson's disease program. And then in the future, it's really focused on validating the efficacy in patients. So this is an overview of our target product profile. A lot I already covered. So the goal is to treat synapse loss and synapse toxicity. In Alzheimer's, it's finding the right patients, but we do so with an oral drug. And that's a huge improvement over the IV infusions that are available to date, and avoids a lot of the risks of safety like ARIA. And happy to take any questions. I'll be around all day and happy to talk about Alzheimer's disease and our programs.
Thanks, Tim. No, we have Ken Wang next for Atlas Home. Thank you.
I know, he actually
Good morning. My name is Ken Wang from Atlas Exomics. Atlas Exomics is a spatial biology tools company. We help companies derive spatial context from biopsy samples. Our chip supplies spatial barcodes to tissue samples that are mounted on glass slides. And from this, we we create a large data set that's used for helping researchers understand what their samples are all about. As you'll see, our platform is very flexible. It applies bulk chemistries and uses them to derive spatial context. What I'm going to cover today is the opportunities, the unmet needs, as well as the technology we've licensed from Yale to advance and propagate the next generation technology. The rapid growth that's expected for spatial biology tools is shown in this figure right here. The market is about $300 million. Today, it's estimated to grow about 30% per year. It'll be $1 billion market in about 5 years, and that's attracted a lot of competitors. So when we considered this opportunity, we said, how can we avoid competition? How can we go and commercialize our business in a space that's unoccupied? When we looked at the current occupiers of the space, spatial biology, we saw transcriptomics and proteomics. And we started looking, where else can we go to avoid competition? And we found that epigenomics, which is a different space, offered a lot of opportunity. Epigenomics in particular is attractive because people are trying to delay aging, right? They're trying to delay diseases of aging. And for that reason, it's attracted a lot of funding. As well, the NIH has boosted its funding tremendously. So we looked at this and said, all right, what are the unmet needs for this space? And we determined that people that are using spatial biology tools are very demanding because they are scientists looking for many different layers of information about the omics. And today, they're only looking at a finite number of targets. It lacks sufficient resolution, and instead of putting together the omics, the information from different omics, epigenomics, transcriptomics, and proteomics, for valuable tissue sections, they have to do them in adjacent sections. As well, there's low throughput. The throughput can be limited to only a sample per day, and it's difficult to analyze and interpret this data. This is a very important factor because, in our own experience, in analyzing the data, it took us as long as 2 months to analyze data from a given data set. And we found that over time, we were able to decrease that time from 2 months to 2 weeks, and our target is 1 day to analyze the data. It's also expensive. Some of our competitors, they have machines that cost as much as $300,000, $400,000 to run spatial omics. And we found that we have an advantage because we can run our assay without requiring a large capital investment up front. We licensed a technology from Yale to meet some of the unmet needs of this space. Yale professor Wang is a member of the National Academy of Inventors, and the platform you see here is useful because it's the same biology platform that can be used to generate omics data from different layers of the regulatory network. You can do whole transcriptome and protein. You can look at the epigenome, which is an unoccupied space. You can also look at co-profiling, which is 2 omics analyses at the same time. What's special about this platform is that the hardware and microfluidic chips can be used, and you can use them to analyze each omics and use the same platform. It's not a conventional technique because most of these other techniques require specialized instruments for each omics. So the platform provides us a special way to get started in the marketplace because the epigenome, as you'll see in this chart, is unoccupied. We are able to start with a new platform that no one else was offering in this competitive space. The other advantage of the platform is that it requires an NGS and chips. The chips we can engineer over time to deliver higher and higher price performance. Right now, we're in the marketplace with cancer centers. We're delivering, performing on a cancer center of over $300,000. The serving major pharma, Department of Defense, represents the largest opportunity. We're working with our collaborators at NTI and also researchers from TBI, Alzheimer's, and aging. We've raised $11.6 million to date in a Series A. We're looking to extend that raise with a $6 million extension, and that round is being led by Spring Mountain Capital. Thank you for the opportunity. I'd be happy to speak to any potential investors later on this day.
So next we have Bexord, Zvonimir, and I'm not going to try the last
You'll want it, right?
Hello. The human brain is the most complex structure in the known universe. With 200 billion cells and 600 trillion connections, it is one of the greatest mysteries of our time. Bexord will be the company that gets to understand the brain. We want to do this because by 2050, we want to develop therapies that will treat more than 500 million people. Currently, pharma uses oversimplified models to understand the brain, and this leads to hindered development of new therapies. So simply put, you can't fix something if you don't know what's broken. So we at Bexord, we turn brains into discovery machines. And the way we do this is that we take dead brains and we revive all cells in the brain and we keep them functional for days at a time. So this allows us to ask experimental questions that are otherwise impossible. And it also allows us to understand the brain at a level not seen before. How we do this, for example, we can take small or a large molecule. We can expose the brain to our substance, and we can study pharmacokinetics, biodistribution, MOA, and PD in hyper resolution. So, for example, we can look for biomarkers fairly easily because the only source of variability on our platform is the brain. And this is because we deconvoluted the whole technology by building it in-house. It has three key components. It's the device that receives the brain, the artificial blood or the perfusate that goes through the brain and reboots all these cells. And then the third piece is the software that actually runs the whole thing, so you don't need to do much. And also, it creates a lot of data. So basically, what we ended up doing is that we created a platform that allows you to study the brain at a really unseen level. We did this at Yale. It took us more than 7 years to get this done and a lot of funding. And this resulted in a couple of landmark publications that validated our technology. We are now an early stage startup going into round A, and we are looking to expand our partnerships with pharma industry and expand our capabilities. For us, the external review has shown that our platform is highly differentiated from anything else that's out there, and we were excited to learn that pharma experts would like to work with us and partner up. For the next 5 to 7 years, we are looking to kickstart our internal drug development by 2027, 28, and by 2030 we are looking to have a robust pipeline in-house that will enable drug discovery and development in CNS space. This is the core team that does all of this amazing work. We are currently at 15 people. And it's a diverse group of people from scientists, physicians, engineers, business people that are led by co-founder Nenad Sesnak, who is a PI here at the Department of Neuroscience, and myself, co-founder and the CEO of the company. Beyond the core team, we actually brought the experts from different fields. So we covered drug discovery, drug development, neuroscience, and entrepreneurship, because we want to grow the company as quickly as possible, and we want to mature the technology to a point where it can allow us to understand the brain. Thank you.
All right, next we have Premal Patel for cell and gene. So if anyone wants to sit, the overflow room is room 4200. Just so we can come in. There is some seats in the front. I can give up my seat here.
Yeah, I would like to sit.
Fantastic. Thank you. I'm Premal Patel, CEO of Cell Infinity Bio, a Yale startup. We're about a year old. We essentially, at Cell Infinity, use directed evolution to identify and develop cell therapy, CAR Ts and CAR NK therapies that can tackle solid tumors. In terms of the core technology of where we're at, we've essentially developed a best-in-field cell evolution platform that allows us to do whole genome gain of function as well as loss of function and CAR engineering processes. And by that I mean we generate isogenic cells identical in every way except one gene is either upregulated or downregulated, times 10,000, all genes within the system, so that we can actually maximally interrogate the overall sequence space and identify genes that would be important. We select genes that would tackle solid tumors, cure solid tumors across a variety of different models, and they go on to become our clinical products. And in terms of where we are with the clinical products, we've identified a lead CAR T with a novel antigen with unique modifications that allows us to interrogate solid tumors, with first patient dose anticipated in 2025. In terms of our overall company, we've raised over $13 million in seed funding, including from a pharma partner, and we are fully operational in terms of labs, with a large lab in the New Haven area and a smaller lab in the West Coast, the San Francisco area. We seek Series A investors and partners for our next steps, which is really clinical development for us. In terms of the overall management team, the company was founded by C.D. Chen, who is a Yale professor within the Systems Biology Institute, a prolific author with Cell Science Nature papers. Several of these are foundational technologies that we've inherited, that we've been licensed via Yale Ventures to become part of Cell Infinity Bio. And the other members have significant C-level experience, chief-level experience within prominent companies, including Liao Immunopharma, Juno Therapeutics, Kite Pharma, as well as Belgium. So an experienced management team, and in aggregate what we're working on is a solid tumor problem. We see CAR T therapies have revolutionized liquid tumors with durable remissions, but they are generally ineffective in solid tumors. And the problems are really multifold, and by that I mean there is inefficient manufacturing initially, and even when the cells go into the bloodstream, they may not infiltrate the solid tumor environment. There's an immunosuppressive environment that prevents T cell from proliferating. Overall, CAR Ts have low potency in solid tumors, and the CAR Ts don't persist. So multiple problems in the field, the state-of-the-art in solving these problems are inefficient in that they are one gene at a time modifications based on, number one, and number two, based on limited biology, spectrum of biology that we know, so that many companies are interrogating the same genes, PD-1 deletions, for example. Our approach is fundamentally different. We use massive parallel engineering to interrogate the entire genome, interrogate 20,000 genes at a time, using multiple different modalities, including loss of function engineering, such that each cell harbors one gene that's deleted, allowing us to interrogate 20,000 deletions. Our winners are what we call endomods. In the second set of screens, we do gain of function screens so that we overexpress one of 20,000 genes and identify those that are winners, and we call those hyperboosts. And the last engineering platform that we have is a CAR engineering, where we modify CARs, their signaling domains, etc. So this allows us to interrogate a very large sequence space, and then we select for the winners in a series of experiments that are relevant towards solid tumors. So identify those cells that are persistent or that can infiltrate a solid tumor environment, that can replicate in an immune suppressive environment, etc. And so in terms of the actual data, an example is the engineering assay to identify winners that are persistent. So you see one of the problems with T cells is that when they are challenged with tumor cells, they can kill the tumor cells for a period of weeks. Afterwards, there's a feedback cycle that Mother Nature has created to prevent autoimmunity that results in exhaustion, dysfunction, apoptosis as T cells die after a few weeks. So in order to circumvent that, what we've done is essentially incubated a pool of our diverse T cells serially with cancer cells in a very canonical, typical serial killing assay. And what we find is that, of course, our cells are bar-coded, so we begin with thousands of unique starters, and then over time, T cells exhaust, they're lost in the overall system. But what we find in our experiment, in our library, is that a few of these cells are alive, remain, kill cancers for a period of months. We identify these genes by sequencing, and they become part of our clinical products. So an example of a clinical product is we took a typical CD19 CAR T and modified it with our gene of interest and identified winners. So the next step for us is going into the clinic with these products and raising a Series A, which will allow us to do GMP manufacturing, clinical trials, and build out our system. Thank you.
So next is Amy Liao from Curagen. Amy?
Yeah, hi, my name is Amy Liao. I'm the co-founder and CEO of Curagen. Before Curagen, I was the co-founder and CEO of GeneWiz, a genomic service company that was later acquired. So my decision to start another company is rooted in the desire to tackle the delivery constraints that many people face when trying to unlock the potential of genetic medicine. So let me start with some basic facts about Curagen. We were founded last year, and we got our seed round closed last fall. And we have licensed technologies from this university and have already assembled a very complementary team, and also got some encouraging results that are embraced by the patient advocacy groups. So over the last decade or so, it's becoming increasingly more enticing to develop therapies based on genetic medicine. On the payload side, there's tremendous progress made especially over the last few years. However, on the delivery front, the two main approaches are viral vectors and nanoparticles, both of which have major limitations. So at Curagen, we try to tackle these limitations by developing a STAP platform. And STAP, it stands for Stimuli Responsive Sequence Engineering Platform. With this technology, the genetic medicine payloads will be chemically modified, and they will be able to penetrate cells through a non-endocytotic pathway. Once inside the cells, the STAP moiety would respond to the cellular stimuli and gets cleaved off, and this will release the payload to its native function. So I mentioned earlier that the delivery to brain is particularly challenging because the viral vectors have the technical limitations and safety concerns, and the nanoparticles are simply too big to travel through this narrow extracellular space in brain. Our STAP technology can overcome these limitations. STAP CRISPR gene editor is small in size and makes it optimum to travel to distribute in brain. And we have shown, both in vitro and in vivo, that our STAP can deliver CRISPR into cells and into mouse brain. So with this capability at hand, together with my co-founders at Yale University, we tried to explore the possibility of tackle Angelman syndrome using STAP CRISPR technology. So one idea to develop therapy for Angelman syndrome is to activate the paternal gene. There's already active clinical trial ongoing, which showed that the ASO treatment targeting the imprint area can improve the patient function, patient symptoms. But there's one major limitation to this approach because the ASO suppression is transient. So when we use genome editing technology, it has some great advantage over ASO because it's permanent and it's more specific. We're able to show that using STAP can deliver the gene editor to mouse brain, and it leads to the activation of the gene. And the biochemical analysis showed that the gene has been consistently and persistently expressed. And the neurological functions got improved too. But these videos will be more demonstrative. The Angelman syndrome mice, when treated with STAP CRISPR, their motor activities are almost as good as the wild-type mice. Yeah, so we believe our technology is truly differentiated and overcomes many of the limitations that I mentioned. And we're very fortunate to have assembled a team that's very complementary with business, science, engineering, and clinical expertise. So to conclude my presentation today, at Couragen, we're building some first-in-class platforms to address the delivery constraints for genetic medicine. We have a great team and great POC data for our lead program. And we believe our technologies will have the potential to enable other genetic disease, other genetic medicine assets as well. 2 seconds to go.
So a couple of things. One, we're working on the temperature, as you can see. We're going to work on that during the break. This is the last presentation in the first hour. After this presentation, we'll be resuming at 10 a.m. 10 a.m. So last is Don Wang from Enrich Biosystems.
Hi everyone, good morning. I free you up in 5 minutes. My name is Dong Wang. I'm co-founder and CEO of Enrich Biosystems. It's my deep honor to be here today as a CI and Armstrong Venture portfolio company. Today I'm going to talk about our innovation, which is microfluidics-free cell discovery platform. T-cell therapy trains the patient's own immune system to eradicate cancer cells. It has shown great power to kill cancers in the past decade. However, the high cost of this therapy is still a huge problem. It costs not only by the downstream process, which is manufacturing, but also by the early stage on how to identify optimal T-cell. In vitro selection of optimal T-cell is such a critical step to increase the success rate of in vivo study. It requires longer co-culture time and optimal effector and tumor ratio, as well as a desired microenvironment. However, existing microfluidics-based technology are not meeting those criteria. Here I'm introducing our Trovo device. It's a benchtop instrument developed by Enrich team. It has the capability to turn your 6-well culture plates into 24,000 micro-well plates within a minute. From there, you should be able to seed your T-cell and tumor cell with a ratio of as you desire and conducting the co-culture experiment for a long period of time. You will be not only be able to observe, but also analyze the kinetics of killing and proliferation over time. And at the end, you will be able to capture, isolate your clone of interest to move down to other downstream study. This is the way we think we're going to connect the dots between in vitro and in vivo cell discovery. Our biggest differentiator is microfluidics free. And we are winning on 4 major aspects compared to other technologies in the market. We at least 10 times higher screening throughput. Unlike other company does 1:1, 1:3 tumor and effector ratio, we have achieved 1:30, 1:50, even chronic antigen exposure. Unlike other company does can handle only up to 3 days of co-culture, we have achieved days, weeks, and months in our lab, and also on the collaborator side. With that being said, the phenotype recognition Trovo will tell you not only the cytotoxicity of T-cell clone, but also have a measurement of proliferation and persistence of those clones. Our beachhead application is a T-cell therapy discovery. We also have an eye on other application to address other markets, such as patient cell line development, as well as sample processing for single-cell mix study, because we have unparalleled capability to dealing with adherent cell and a dirty tissue sample. Enrich team consists of a startup operator and a scientific innovator. We co-founders previously involved 2 other startups with good assets. Right now, we backed by a great set of institutional investors as well as angels from all over the world. Since we closed Series C round last year with 30% oversubscription, we have strengthened our KOLs with Dana-Farber, Yale, UPenn, and MD Anderson. We invested in system integration, and we're turning our beta system to first-generation Trovo product. This year, with projected Series A financing, we'll be further develop our software, which is a machine learning module. We can even more precisely process the experiment and has a better readout for phenotype. And we're also going to be using the Series A round financing capital to build full-scale manufacturing and commercial operation. We believe better cells, better data, better biology will lead us to better cures. Thank you for your attention. Looking forward to connecting offline.
Thank you very much. Okay, so we are at a break period until 10 a.m. and we will start at 10 o'clock sharp. So if you want to go get some refreshments and just enter back here like that. Thank you.
We're good, right? I'm happy to continue. Whatever you want. Yeah, slide should be here. It'll be there. Just like everybody else, my fully processing is so smooth. The slides.
I know, usually I'm like,
I'm Frank, by the way. Nice to meet you. So we're going to get going in about 5 minutes, so please find your seats when you can. Thank you.
on one. I don't think chemistry of it. I never had time to do it. I yeah, just some of the pieces to Vineyard. So now it's on. That's it. Getting in the brains. So I'm just gonna, there may be a bit of as we do that interface, we'll just make sure that like when you come up all the can I see it with the note? That's great. That takes a ton of I can see it like that. You don't want that. And the current issue is that when I'm projecting it, hey Bill, are you presenting?
All right, if you can start finding your seats, that would be appreciated, please. 2-minute warning, find your seats, please. Again, can you please find your seats? Okay, we're going to get going, please. So can you please find your seats? Okay, I'll let them know. Perfect. All right, thank you very much, everyone. We're going to get going, so please. Please find your seats. Please find your seats. Thank you very much. Please. All right, everyone, we're going to get going. We're on a tight deadline here. If I can get your attention, please. Thank you. Please find your seats. Please find your seats. Thank you. All right, thanks everyone. Thank you. Okay, thanks very much. We're going to get going again here. Okay, people, please, we have, thank you very much. We have another exciting group of companies. We're good? Okay, thanks everyone. Welcome back. We have another exciting group of companies. Just so you know, the slides, all the presentations are on the innovation website. So if you want to follow up with companies and you missed them here, you can go to the website, you can get their contact information. So don't feel pressure to track them down. If you can't make that, just remember everything's on the innovation website. So we have next presenting Freedom Biosciences. Dina's going to present. Thank you.
Thanks so much, Frank. Hello, everyone. My name is Dina. I'm the co-founder and CEO of Freedom Biosciences. Very excited to tell you more about the company. So we are focused on solving what we believe is one of the largest problems facing humanity today. It's mental health crisis. There's over 300 million people diagnosed with major depressive disorder, and despite the fact that there's over 50 antidepressants on the market today, a third of this population, or 100 million people, are still treatment resistant. So as some of you may know, ketamine is a truly transformative mental health treatment. Thomas Insel, who is the former NIMH director, said that ketamine might be the most important breakthrough in antidepressant treatment in decades. And it was actually here at Yale, led by Dr. John Crystal, who's just right here in the audience, and Dr. Rob Berman and their collaborators, that they showed that ketamine was an antidepressant in the 90s. There are many reasons why ketamine is so transformative, but one of the biggest things is it's the fastest acting antidepressant. Many patients get relief within hours, and you compare that with SSRIs, for example, that takes weeks for patients to get relief. And that is really what's responsible for such a huge explosive growth in the number of ketamine clinics over the last 8 years, the number of those almost 10x'd. So this really brings us to our lead program, Freedom 001. Again, it's another study that was run here at Yale, led by Dr. John Crystal, who's my co-founder, in Freedom Bio, that showed that the combination of ketamine and rapamycin had a much more durable effect at the 2-week point. So if you look at the graphic here, in 2 weeks after treatment, and this is in 20 patients in a crossover design study, in 2 weeks, the response rate for ketamine alone was just 13% compared to over 40% for ketamine and rapamycin, and remission rate for ketamine alone was 7% compared to almost 30% for ketamine and rapamycin. So these are truly incredible effects, and this is potentially means that Freedom 001 might be one of the most effective treatments for depression. So what does this mean in practical terms for patients, the healthcare system, and payers? It means that we might be able to reduce the number of sessions that patients need to have by 3. So for every 3 ketamine or Spravato, which is S-ketamine, Janssen's product, there might just need to be 1 Freedom 01 treatment. That means that it might be 11 times cheaper for the patient to go through the treatment than ketamine, and 3 times less expensive for both the patient and the payer. There's also additional benefits outside of the financial benefits. It's higher convenience, lower abuse liability, improved patient safety, and adherence. So you might be wondering about the commercial viability and the opportunity here. So Freedom 01's closest proxy is Spravato, which is, again, Janssen's product that was approved for treatment-resistant depression in 2019. Based on the latest Q1 2023 numbers, Spravato is now the fastest-growing program in Janssen's portfolio. There was an 87% increase in sales from Q1 of 2022 to Q1 of 2023. It is now on track to become a blockbuster next year. So I think as the first approved psychedelic on the market, this really shows that psychedelic therapeutic can be truly a commercial success in big pharma terms. So zooming outside of just Freedom 001, Freedom Bio's platform includes combination therapies of next-gen antidepressants such as ketamine, other psychedelics or 5-HT2A agonists, and pathogens in combination with pharmacological enhancers to improve on the safety and the efficacy of the next-gen antidepressants. And we have other preclinical programs with promising animal data, including a non-hallucinogenic psychedelic. We have an incredible team. Dr. John Crystal, who's my co-founder, he's the chair of psychiatry at Yale, and he led both the 01 discoveries. David Hoff, our CMO, he led the Spravato program for 7 years at Janssen from phase 2 to commercialization. And Dr. Rob Berman, who's the co-founder and former CMO of Biohaven. We're currently raising money, so if you're interested to speak to us, please get in touch. Thank you.
Okay, great. We have Modified Bio. Okay. So we're good.
Wonderful, thanks so much. I'm a professor of radiation oncology and co-founder of Modified Bio. Really excited to tell you about the story. So for this slide, it's just the high points. We're doing something that no one is doing in the oncology space right now, which is exploiting cancer-associated DNA repair defects by DNA modification. We have a strong team, and we have a lead asset that's headed into the clinic. We're candidate nomination imminent in the next 2 quarters. We hope to be in the clinic by the end of next year. We are not only a lead asset focused program, but we also have a platform technology that we can apply to many different DNA repair defects. And we're currently raising the seed 2 round, which we hope will support us to nominate that candidate and close our Series A later this year. In terms of the team, I'm one of the co-founders. We also have Seth Herzon from the Department of Chemistry at Yale, Kingston Lin from the medical school, and my longtime business partner, Kevin Rakin. Kevin and I have started a few companies you may be aware of. I'm one of the co-founders of Cybrexa Therapeutics as well as Alphina Therapeutics, and this is our next big thing. For the team, just want to show you those logos. What's really exciting for a seed stage company, we've been able to recruit people from publicly traded companies to come to New Haven to go work for us. And similarly, we have an advisory board on the SAB of KOLs crossing the bench to bedside space and the glioma and the non-glioma realms. So what exactly are we doing? So in the DNA repair synthetic lethality space, we'd argue that almost everyone is indirectly targeting proteins. They indirectly target those proteins to create complex DNA lesions that DNA repair defective cells cannot resolve. We're going back on that central dogma and actually modifying the DNA directly. So I'm in clinic today radiating patients, and we radiate the tumor DNA, and it works really well, but the therapeutic index is the problem. And so how is Modify going to solve this? Well, we have a core technology which we call DNA modifiers. These are like ADCs or peptide drug conjugates, but very simple orally administered molecules consist of a vehicle and modifier. These release the modifiers, bind to the DNA, but we iteratively refine them so normal cells can rapidly remove them, but cancer cells with specific DNA repair defects are unable to resolve them and subsequently die. We have a lead program just at a high level today, MGMT, called methylguanine methyltransferase. This is a gene that's silenced in many different cancers. Temozolomide and conventional chemotherapies work really well in this space, but they acquire resistance via mismatch repair mutations. And the mechanisms on the right, what I'll tell you is if we can find a DNA modifier that can exploit the MGMT defect and be impervious to tumor-associated mutations, we'd have a therapeutic win. That's exactly what we found. So this has been an academic collaboration that moved into Modify. We published this paper in Science just last summer. And we showed here, I'll just draw your eyes to the green and the yellow curves here. You can see the MGMT-dependent interaction impervious to mismatch repair mutation status. Very, very nice effect in vitro that we also see in vivo. So I'll show you here on the next slide, this is a PDX model with our genotype that's resistant to all the standard of care therapies. Intracranial xenograft model, 3 cycles, monotherapy efficacy, 8-fold improvement in overall survival. I've been working in the lab, running the lab for about 10 years. We haven't seen data like this in quite some time. We, in parallel as a clinician scientist, working with my SAB to develop our clinical trial, because we will be in the clinic in about a year. You can see here precision oncology basket trial design, which we're very excited about. And then we'll just end here with our drug development strategy overall. As I mentioned, what's unique about the seed stage company is we have a candidate nomination imminent, and that will put us in the clinic by the end of next year. We completed all this work with very financially disciplined strategy with a seed 1 round of $6.4 million and a phase 2 SBIR. We're now raising the seed 2, currently at $4 million, but we're extending it because of the investor interest. So certainly reach out to me if you're interested. And we hope by closing that seed 2, we'll nominate our candidate and be able to close our Series A by the end of the year. So just leave you this as a visual. I'd say we're very excited about an entirely new way to target DNA repair defects in cancer based on high impact cutting edge science at Yale. And we're very close to the clinic, and we're excited to extend the platform out to many, many different DNA repair defects. And so definitely reach out to me if there's any interest. Thank you.
Okay, great. We have an overflow room, again, in room 4200. If you can't find a seat, you want to sit, you can still listen and see the presentation. Next, we have, who's next? Bill, right? Normunity, Bill LaRochelle.
This is forward. Okay. Okay, great. So I want to thank everyone and say good morning to everyone for coming. What I'd like to do this morning is to present to you a company that was just founded last year called Normunity. Now, part of the problem that we're trying to solve and the foundational question for Normunity is why do so few people, or why do half of the people treated with immuno-oncology therapeutics not respond? The idea then is that, and I would like you to imagine a tumor in situ, the idea is that a tumor is surrounded by a barrier, and what we want to do is remove these barriers. We want to remove barriers to T-cell infiltration. We want to remove barriers to immunotherapeutic approaches. And just from the 40,000-foot view, Normunity is seeking to normalize the immune system. And as you can see here, we have the typical cold tumor or the immune desert, followed by the hot tumor or the immune responsive tumor or the inflamed tumor. And Normunity is seeking to remove these barriers by novel targets discovered by Li-Ping Chen's lab. Now, this is a much more detailed view, and you can see on the right-hand portion of this slide, the two panels with the yellow staining shows pretty potent T-cell infiltration across representative samples of more than 2,000 multiple tumors across many different cancer types. And then the question becomes why in the two left-hand panels there are no T-cells. What is keeping the tumor microenvironment to be a immune desert? And then in some cases when you even see inflammation or the presence of checkpoint inhibitors that indicate a potential checkpoint inhibitor therapy, you see no immune responses. And this just gives you an idea of some of the numbers. More than about half or more are served by currently available therapies, and the huge market opportunity is the right-hand half, which the fundamental question we're trying to answer is what prevents the T-cell infiltration. And indeed, even some of the currently served or available therapies are either refractory or don't respond. Now here is our lead hypothesis. We would like to basically target substances within the immune environment that normalize T-cell infiltration. And these are targets discovered by the Chen lab. Typically, these targets we call immune disruptors. They're tumor specific and they're not in the normal tissue. Many of these are fertility type of antigens, or they're expressed in testes, etc. But again, the fundamental hypothesis we're testing is that the infiltrating tumor cells would be vital to the immune response. Now, I showed you why we're doing what we're doing. I want to show you who we are and how we're going to do it. So this is our team. Li-Ping Chen is the founder. Rachel is our CEO. I am the CSO, and Melinda Merchant is our clinical development person. Saul Fink is in charge of non-clinical development. Olga Granitova is our chief business officer, and the way that I wanted to present this slide as well is to show you that I believe we have a truly outstanding team. We have a track record of success. If you look at it, we have greater than 30 regulatory approvals. Again, these are just some of the drugs that we've all touched through our work, and again, we're involved multiple big pharma experiences, Bristol Myers, AZ, Roche, Pfizer, Merck, just to name a few. So our platform is The platform developed by Liping Cheng. Right now we're working on what we call a T-cell mobility platform, and this is a platform that selects for T-cells and how they move in and out of tumors. Liping also has 3 proprietary platforms that we're working on. We're very excited about some of those targets, and we're going to be able to work as they become much more mature. So a very robust pipeline of targets. So in the end, how are we going to do this? So what we're doing right now is we're identifying novel targets with Liping. They're also coming out of his lab and his high-throughput screening platforms. As I mentioned earlier, he's generating other additional targets, testing the hypothesis in animal models. So we have a very strong collaboration because we're together. We're rigorously validating the targets to understand their MOAs. Liping and we together produce proteins. He does very often murine antibodies for proof of concept. We do immunocompetent, non-competent animal models, and we do further testing in collaboration while very often identifying biomarkers. And then what we're doing, or what Normunity is doing, is developing fully human antibodies with Normunity and moving them towards clinical development, precision markers and precision medicine. So what I want to leave you with is this slide. So the corporate highlights for Normunity is we've raised $65 million in a Series A last year. We have a fully operational discovery unit in 520 at Biolabs where we can collaborate very robustly with the Chen Lab. We also have corporate offices at 1 Beacon Street in Boston. We're a very lean company with 14 full-time employees. I showed you we have a very strong clinical development team pushing toward the clinic, and we have a world-class scientific advisory board. I just wanted to shout out people like Mario Snoll from Yale, who's been great at helping us, as well as Roy Herbst. And in the end, we're Normunity. I showed you why we're doing it, what we're doing it, who we are, and how we're doing it, and we're www.normunity.com for more information. Thank you.
Okay, we have Amy Divaraniya. Told you I was going to mess it up. My apologies. Uva. And again, we have all the presentations are on the Summit website. So if you do want to get in touch with anyone, overflow room is room 4200. So if you do want to see, you don't see one here, you can still hear and see. And that reminds me, if you have cell phones, please put them on silent if you can. That would be wonderful. We're good?
Okay, great. Thanks. Thank you. Hi, everyone. So 87% of women do not have a 28-day cycle, and yet all protocols and products use the 28-day cycle as the foundation. Sure. To me, this is a clear call for us to redefine the normal woman. My name is Amy Divaraniya, and I am the founder of UVA. Unfortunately, I am part of the 87%. I have always had an irregular cycle. When my husband and I wanted to try to have a baby, we expected the road wasn't going to be super easy. However, nothing could have prepared us for the heartbreaking and devastating 18-month journey we had to endure. The worst part, luckily my story had a happy ending. However, it opened up my eyes to just how many big gaps there were in women's health. The primary one that really stood out to me was how little information women had about their fertility, specifically regarding their hormone levels. Our hormones dictate everything in our health, and yet when I was in my journey, it felt like somebody was holding the key or the password to the secrets of my body, and I just wasn't privy to that information. I wanted to change that. Despite having a PhD and being trained in data science, I learned very quickly that I just didn't have the information I needed to advocate for my health. So what was the solution? The only solution available to me was to go into the clinic every day for an invasive blood draw and then wait for those results. Now let's walk through that. Not only do I have to go into the clinic every single day, I had to get a blood test, and then I also had to find a clinic that would treat me, and most importantly, a clinician I would trust. What if I didn't live in the metro area? What if there was no clinic nearby? Did I really have the bandwidth to travel hours to get access to this information? There had to be a better way. So I set out to build a platform to provide women with that hormone data in the privacy of their own home, in real time, and most importantly, non-invasively. We built an end-to-end platform that consists of 3 components: a urine test, an mobile app for users, and then a web portal for clinicians. UVA is very easy to use. A woman simply provides a urine sample on an UVA test, scans it with her phone, and within minutes, she can view the results in her app. Simultaneously, her clinician can view the hormone data directly in their web portal, enabling them to take clinical action as necessary. While the hormone data is informative for both the patients and the clinicians, what's really empowering for UVA is the unique data set that we're creating. We have curated a completely unmatched data set that marries daily hormone levels with self-reported symptoms and treatment protocols. Our machine learning algorithms learn the nuances of every woman's cycle and then builds a predictive model to help achieve her health goal. Yes. So the way that this works is we actually have a completely personalized experience. We learn what every woman's unique hormone baseline levels are, and then we detect fluctuations by comparing to that. But we're not doing this alone. We have partnered with some of the leading clinics and clinicians in the space to actually challenge the status quo. For instance, last year we submitted and presented 2 research articles and posters at leading fertility conferences. And what's really exciting to me is that with all of our research, we're starting to redefine what advanced maternal age means. What is a geriatric pregnancy? What does irregular hormones or irregular cycles mean? And what does a hormonal imbalance truly look like? With the technology and this invaluable data set that we're curating, we're starting to really revolutionize women's health. And the best part is we're just getting started. So I welcome you all to join me in redefining the normal woman. And if you're interested in learning more or working with us, please feel free to reach out. Thanks.
Okay, we have Bob Linke from Osmo Therapeutics next.
Okay, so where is my
Welcome, everyone. Thanks for coming to Osmo's presentation here. I want to thank Yale Ventures for inviting us to present. We are a development stage pharma company developing technology out of Dr. Barbara Ehrlich's lab here at Yale to deal with a very big problem in oncology, chemotherapy-induced peripheral neuropathy. We talk a little bit about the team. You see Dr. Ehrlich at the center have assembled a great development team here. Together we have over 125 years of drug development and commercial experience that includes oncology products, CNS products, and repurposed pharmaceuticals. Let me talk a little bit about CIPN. Essentially, we deal with the off-target toxicity of many chemotherapy agents. One of the biggest of those are the microtubule-based agents, where our drug works. Essentially, we deal with the off-target toxicity. Our initial focus are the taxanes. We estimate there's about 360,000 patients that get taxanes every year in the U.S. and the EU. And the initial focus is breast cancer. We're up to 80% of breast cancer patients who get treated with taxanes develop some form of peripheral neuropathy. It starts out as tingling in your hands and feet. Suddenly you can't tie your shoe. You have trouble walking. It can be disabling. And why is it such a big problem? There are no approved disease-modifying treatments today. We hope to be the first disease-modifying treatment available as we enter the clinic. We're going to file our IND this month. So a little bit of our product. We deal with that off-target toxicity that damages neurons. Essentially, we protect neurons from this intracellular calcium surge that starts a cascade of neuronal destruction. Pre-treatment, you would use our drug, administer our drug 30 minutes before each chemotherapy infusion to protect neurons, and we hope to also improve outcomes, because half of the patients that develop, in breast cancer, half the patients that develop CIPN, it's so bad that they have to have their dose adjusted to deal with the disease. So a little bit about our product. We're a patent-protected novel formulation of lithium chloride that really enables use in CIPN. As a result, we'll follow the very efficient 505(b)(2) pathway. and high confidence of clinical success given Dr. Ehrlich's work in mouse models of CIPN, as well as a second potential indication, cognitive impairment. The same damage that happens in the periphery happens in the central nervous system as well, causing cognitive impairment. We understand the mechanism of action very well. We actually have some clinical data to support this, and 50 years of experience with the active agent. And most importantly, as a repurposed pharmaceutical, our IV formulation creates significant barriers to generic intrusion. A little bit about the market. Those 360,000 patients translates into about a $5 billion potential market. If we could address 25% of that, it's a $1.2 billion sales opportunity. We think that's relatively conservative because we haven't included China or other microtubule-based chemotherapy agents in that number. We talk about the market research with oncologists and payers. We did that to learn more about the problem and get feedback on our product. We talked to oncologists at leading U.S. cancer centers as well as payers that cover 100 million lives here in the U.S. They confirmed, got to have preventative treatments here. There's nothing out there other than changing the dose. There's nothing available in terms of a disease-modifying product. We reviewed our target product profile with the clinicians and payers. They were very enthusiastic about our product. If we could reduce CIPN even on a relative basis, 40 to 60%, they would use it with all of their patients. And we also took them through the use cases of using the generic lithium carbonate product versus our IV product, and to a clinician and payer, they said they wouldn't touch the oral product given toxicity, side effects, the therapeutic index, and the cumbersome nature of using the oral product. Patents, we have patents issued in US and China, pending in the EU. We have plans to file new patents around manufacturing methods that we just discovered as we finished our initial manufacturing work, as well as new patents once we enter the clinic around dosing. A little bit about our development pipeline. As I said, finished all of our IND enabling work, going to file the IND this month. We're very excited to be in the clinic. We're raising $5.5 million to fund our phase 1 study, as well as two projects, our tox study and manufacturing for our phase 2 program that we hope to begin in the middle of 2024. It includes a study in metastatic breast cancer patients here in the US, as well as an early stage study in taxane-treated breast cancer patients in the EU. Talk a little bit about our financing. We're raising $5.5 million to fund us to the phase 1 and be phase 2 ready by the middle of 2024. Our previous financing was led by COAX Investment Partners. This is a group of COAX founders. Rob Berman's our chairman. Vlad Kork, the CEO, is an investor, as well as one of the Biohaven board members. Again, this will fund all of our IND enabling work to get us to phase 2 ready, middle of 2024. And we have initial commitments to the round, term sheets available for review with interested investors. Thanks for your time and attention. Would love to talk to anybody that would like to learn more about our development program. Thank you. Thank you very much.
Okay, Susan Froshauer from Pangolin Therapeutics. Susan?
Okay, go away.
Just advance.
Okay. Yeah, I got it. Good morning. I'm delighted to be here to present our company, Pangolin Therapeutics. We are an early Yale startup company dedicated to a chemistry platform delivering a brilliant novel class of therapeutic agents we call pangamirs. This technology was invented by our co-founder, Andrew Moranker, who is a professor of MB&B at Yale, a biophysicist, thanks to our co-founder, Enrique Alvarez, who brings over 25 years of experience in IND enabling work to the company. We are well along in developing our pangamir programs towards INDs. So what's valuable about pangolin is that we target a otherwise abundant class of intrinsically disordered proteins, intractable proteins for drug discovery. Unlike enzymes or classical drug targets which fold and have small binding sites, the IDPs have a dynamic structure and no little binding pockets for small molecules. IDPs cause in disease states the formation of toxic structures that propagate disease. And I think you know proteins like A beta in Alzheimer's disease and alpha-synuclein and Parkinson's disease that are among these classes. What's great is our pangamirs, which are complex structures in and of themselves, they're hybrids between peptides and small molecules. They have the ability to bind IDPs and prevent their toxic structures from forming. So our approach at Pangolin is to harness a multidisciplinary strategy, lots of different tools to design new pangamirs. And very importantly, we know it works because we have a first-in-class lead candidate for type 2 diabetes that I'll tell you about, 1023. So a little bit about how a pangamir operates. It is also dynamic in its structure. In its compact form, it's able to land on energetically favorably influence the formation of a structure, in this case an alpha helix, and a protein target. The pangamirs themselves in this compact state are accessible for traditional small molecule synthetic organic chemistry, so we can make drugs. So what I want to focus on, and I mentioned, is our most advanced program targeting IAPP, islet amyloid polypeptide, which is a new target to treat diabetes. IAPP is in the case in beta cells when there's an overproduction of insulin. IAPP is also overexpressed and it forms toxic structures. And this certainly is the case in diabetes. So the consequences of these structures, which land on the membrane of mitochondria and form large pores, is the beta cells die. And so we are very excited because 1023, our lead molecule, which engages toxic IAPP structures, inhibits this toxicity, and we believe we have a cure or after a short course of therapy. We think this will compete in the complex diabetes market. So this table summarizes a compendium of data we have on 1023. Most importantly, I'd like you to focus on the efficacy in a really well-trodden animal model of type 2 diabetes. Having efficacy for 1023 is a proof of concept that these pangamirs are drugs. We show membrane crossing and solubility. So what I want to summarize with here is that the value proposition to the investment community. We're here today to launch our seed round, and the value, in addition to some of the things I highlight, are that we have the capacity to build a robust pipeline across multiple therapeutic areas. I've spoken to you about the diabetes opportunity, but we have really great molecules targeting alpha-synuclein for Parkinson's disease. We're very interested in pursuing cancer targets. So we have the team to execute on this program. I've mentioned Enrique and Andrew. I also bring some experience. I'd like to just briefly point out Bob Volkman, who's an expert medicinal chemist out of Pfizer, who has advised us on pangamir synthetic or organic chemistry and SAR. And to the right on the bottom, a really great scientific advisory board. Thank you very much for your attention.
Okay, we have personal therapeutics. Marc Pelletier. Hey, Marc.
Coming, 5 minutes. Marc is there.
It's a great honor to be here to present on behalf of Personal Therapeutics. Personal Therapeutics has 2 drugs that regulate renalase, which is an extremely powerful biological pathway discovered by Gary Desir, the chairman of internal medicine here at Yale. And it's the power of that pathway is that if you activate it, you can treat inflammation, and if you turn it off, you can treat cancer. So our 2 drugs are BP1002, which is a proprietary renalase agonist, initially for acute pancreatitis, And BP2002 is a humanized antibody for the treatment of drug-resistant cancers. We have proof of concept in multiple models for both compounds. Proprietary biomarker assay. It's a druggable target with inpatient validation. We're 12 to 15 months from an IND, post-funding of each project. And we're seeking $5 million to advance the lead compound, the BP1002 to IND for acute pancreatitis. This is not our original slide deck. Acute pancreatitis is potentially life-threatening. Pancreatic inflammation with 10 to 30% mortality and severe disease. The hallmarks are severe incapacitating pain, nausea, vomiting. Hospitalization is 2 days to 4 weeks. There's an increased risk of chronic disease from diabetes, pancreatic cancer, chronic pancreatitis. The clinical and commercial opportunities, well, acute pancreatitis is the leading cause of gastrointestinal hospitalization with 250,000 cases and $2 billion worth of hospital costs annually. And we have a rapid path to clinical proof of concept. The lead compound, well, it targets BP1002, it targets renalase, which is a secreted protein that protects cells, reduces inflammation and pain. Mechanism of action has been determined. Plasma renalase levels are significantly reduced in patients with acute pancreatitis. It's a proprietary small peptide that's easily manufactured through chemical synthesis. We have preclinical proof of concept in several models of acute organ injury, including pancreas, kidney, and heart, and also have been shown to reduce pancreatitis-induced pain. Let's go to the next slide. So this is the tip of the iceberg of a huge body of work. BP1002 decreases histologic injury in a dose-dependent manner, and it's effective as a therapeutic without pretreatment. The tissue not only looks, tissue on the far bottom right there, not only looks better, but the animals are do really well. And the inflammatory cells are significantly reduced. If you look at the neutrophils, they're way, way, way down. Our pipeline is divided into 3 products. We have a renalase agonist for acute pancreatitis. We're seeking $4.95 million to take it to IND. The follow-on indication is acute renal failure. And we also have the possibility of working on viral-induced hyperinflammation. We have the antibody, the humanized antibody for the treatment of melanoma, pancreatic and colon cancers. And importantly here, we have a biomarker that's in development which will help us facilitate patient selection in clinical trials. So this represents turning it on, turning it off, and monitoring it in a clinical trial. These are all key elements in our program. We have a great team. Pharmaceutical executives. This was founded in part through a collaboration with Besser Pharma. They founded Personal Therapeutics with Gary Desir. Barry Berkowitz, sitting right here, is fantastic. And Elliot Olstein, Marc Rothman, Paul McGurk is also on the team. They have decades and decades of experience and brought many drugs to market. On the Yale side, Gary Desir, co-founder, chairman of Department of Internal Medicine and chairman of the board of Yale Medicine. Fred Gorlick, who's a world-leading gastroenterologist. Harriet Kluger, just a really fantastic team of advisors as well. So what we have is a target that regulates inflammatory disease and cell survival. BP1002, our first drug, is we're taking it to IND for acute pancreatitis. It's an agonist, small peptide. BP2002, the humanized antibody, is staged for development of melanoma, colon and pancreatic cancers. And importantly, we have a biomarker to help us with our patient selection. Thank you.
Thanks, Mark.
Oops, sorry. Patrick Jin. David? Is David here? Hi.
Again. Timer there. Thank you.
Awesome. Thank you. Awesome. Thank you so much. Thank you all for being here today, and it's my honor to present some information on our company, Petrogen, where we're focusing on developing novel therapeutics for periodontal disease. So just starts out, I'm very proud of our team we put together. That's over 120 years of experience in pharma and biotech. Everything from starting companies to moving drugs into clinical trials to getting drug approvals. Our team's got at least 10, I think, maybe a few more. We also have expertise specifically on our therapeutic target with the work that Demetrius Braddock has done here at Yale, along with expertise in periodontal disease, specifically with Martha Summerman, who's a former director of the National Institute of Health's dental section. So where does our story start? So work at Dr. Braddock's lab was specifically focused on an enzyme called the NPP1, and specifically the deficiency of the NPP1, which leads to a disease called general arterial calcification in infants. It's a terrible disease. Mortality rates over 50% in the first 6 months of birth. And literally what happens is that there's a calcification in the body, heart, arteries, all sorts of terrible things. But in the work that he had done, he noticed something very interesting. So if you look at these GACI patients, there's material here, this yellow marked out material, is called cementum. And you notice that in GACI patients, that material, that cementum, was much, much thicker than healthy controls, where the material is thin. So why is that important? Well, cementum is the material that holds your tooth and your gum together, right? So your gum actually doesn't attach directly to the dentin to your tooth. It actually attaches to cementum through these Sharpie fibers, and that really allows it good clinical attachment, which is very, very important. In periodontal disease, you have a gap between the tooth and the gum, and sometimes there's not a lot of cementum there. And so that's really an exciting opportunity where Demetrius said, well, I don't have periodontal disease expertise, but let me bring in and collaborate with the NIH. So that's exactly what he did. So here's just some published data. You can see again on the left side here, control, you've got thin layers of cementum, and these are all GACI patients. You see much thicker layers of cementum. So, in my experience, having been in the pharma biotech space for almost 30 years, normally when I see an idea, it's a hypothesis that, starts out in vitro, you do some cell work, you do some in vivo work, eventually you get to the clinic and you really try to prove out in humans whether what you thought was going to happen happens, right? This is the first time where I've ever seen something where the human proof of concept was the idea for the company, which is really super exciting. So now our job was to sort of backfill that data and make sure that our target's really well validated. So here's some work that was done and again published in peer-reviewed journals. Here's a mouse model. You've got on the left side wild-type mice, and you see this thin layer of cementum. And then over here in our missense model of ENPP1 called the ENPP1-ASJ, you see much thicker layers of cementum as well. So, we wanted to do it yet again. You need to validate your target multiple times. This is the orthodontic tooth movement model, another paper published in peer-reviewed journals. And again, left side, wild type, you see thin layers of cementum. On the right side, you see much thicker layers of cementum. Here's a topographical micro-CT, and here's some histology. Again, acellular cementum is a thin layer on the wild type side. And again, on the ENPP1-ASJ side, you see much thicker layers of cementum. So what's really the idea here? Right. So the goal is to really, provide restoration in periodontal disease by dropping ENPP1 inhibitor in the pocket of the tooth and have it a local effect. So this is not a systemic drug. This is a local applied drug. And we'll talk about sort of the benefits of that in a second. So how big is the market? 36 million people in the U.S. alone have periodontal disease. That's about a $15 billion market. Ex-U.S., if you look at just kind of high-income Asia plus high-income Europe, that's another 45 million patients. So the overall market's probably about 35 billion or so. So what's standard of care? If you're going to come into a market, you better know standard of care. That's very, very key. So here, a patient comes in, there's some bleeding, inflammation. So the dentist or periodontist will now take a probe, start measuring sort of the clinical depth in the pocket, so that's measured in millimeters. And then they'll do what's called scaling and root planing. So for almost $400, you go in and they take a probe and scrape away the bioburden, the debris, the calculi. And that actually helps the first time you do it, but after that, really not so much. So if your pocket depth, which is how they measure periodontal disease, is 5 or 6 millimeters, which is considered moderate, sort of borderline severe, you're going to get about 1 millimeter back in terms of clinical depth. Second time you do it, really is not going to work all that well at all. So What they actually also do, in some cases, is add an antibiotic after, and that's a drug actually called Arrestin. It's actually the only non-invasive therapeutic available in periodontal disease today. And so Arrestin is just minocycline in a PLGA microsphere put into the pocket of the tooth. And so that's really just kind of the same modality we're going to do as well. As you can see, the antibiotics don't really work very well, between 1/6 and 1/3 of a millimeter of benefit, so that's really clinically meaningless. So at the end of the day, I'll just jump ahead and kind of give you where we are and where we're going. So we've done target validation, as you've seen, right? We understand the market, the standard of care, the regulatory pathway. We've raised $3.2 million in seed capital to get us to our what's nearly our clinical lead. We're going to declare our clinical candidate shortly, and then move into phase 1 studies, which will be hopefully done beginning of next year, or started beginning of next year, and have data read out about 12 to 15 months later. So thank you very much. Thank you.
Okay. Susan Sobolov from Rigamune. Susan?
There you go. Thank
Timer's
you.
here.
Timer's there. Perfect.
All right, Susan has the last presentation of this block. After this, we will be on a break until 11 o'clock. So do what you got to do and then get back here 11 o'clock sharp. Thanks, Susan.
Thank you. And thank you all for attending and being interested in learning about Rigamune, where we are targeting RNA therapeutics against the RIG-I receptor. Rigamune was started in 2020 by Yale professors Anna Pyle and Akiko Awasaki, who built the foundational understanding of the RIG-I pathway and the activation with our SLR molecules for activating RIG-I. I also want to congratulate Anna Pyle for her recent election to the National Academy of Science. So our platform at Rigamune is bringing together the knowledge, the structural knowledge of how pathogenic RNA bind to the RIG-I receptor and activating it. With the knowledge of this information, we have used the minimal structural determinants to create the synthetic SLR molecules which mimic the pathogenic RNA and therefore are agnostic to the virus that may be present and also do not cause emergent resistance. These molecules are highly potent, highly selective, and we've been able to optimize them for drug-like properties, leading to our lead molecule, RIG-101. Now, most of us know with RNA molecules, a key component is the ability to deliver the molecules. And we have acquired a company in 2022, Subintro, that brought to us respiratory expertise in formulation delivery, and together we have developed targeted proprietary non-LNP formulations. And why is that important that they're non-LNP? For our first target with Gates Foundation against an intranasal delivery for respiratory diseases, one cannot be using LNPs to deliver to the nasal cavity, which have shown to be highly inflammatory. Our non-LNP proprietary formulation has shown to be safe and effective with no impact on inflammatory markers. So with our intranasal delivery, we have the opportunity to prevent transmission and replication at the mucosal surface, the actual site of viral infection. Additionally, with our platform overall, we have the potential to be a platform within a product with activity pan-viral, as well as effects against inducting immune response in cancer. Very exciting to be showing our proof of concept data with the intranasal delivery in a mouse model of influenza delivered in a prophylactic approach. We have shown significant safety and tolerability by reduction in body weight loss,
as well as lack of neutrophil infiltration, both in the lung and the nasal tissue. And excitingly, you can see a highly significant viral log lowering throughout the 5 days of the study, both in the nasal tissue and in the lung tissue. So what we've been able to do with our seed funding, our funding from the Gates Foundation, along with F-Prime and our acquisition of Subintro, we have been able to identify our lead molecule, develop scalable manufacturing, and deliver on key preclinical milestones. The intranasal formulation, the preclinical proof of concept with that intranasal formulation, as well as, very importantly, we now have a proprietary non-LNP RNA delivery platform that works not just for our SLRs, but a broad approach for delivery of RNA molecules. And we are rapidly progressing our IND-enabling studies to deliver, to enable starting our clinical trials in 2024. We are also now kicking off our Series A funding to be able to fund the next level of milestones of looking at and enabling our clinical proof of concept and differentiation. So I'd be happy to speak with anybody and tell you more in details about Rigamune and what we're doing. Thank you.
Thank you very much. Okay, so we're on break until 11 sharp. Thank you.
No, I haven't seen it yet, but it's been super helpful. And everything's set up. And, we got all of our operations.
Don't know anything. And where does that?
So I do nothing in the lab, and we do all of our lab stuff.
I just know it's very confident. Now I see
That's what I called you about. That street, and then give me
asked a lot of questions,
I'm interested in I'm just gonna try. Let's try to do this. Previously, when they were, yeah. That's fine. Come on, Ben. It's sure. Yeah, so take the word, even though I think the hypothesis from Denisovich, right, is that maybe probably not the blacks that are the most it's the beta. So Dennis said, what's the mechanism for and to strip out or piece together the mechanism of the surface and it requires mTOR5 to trigger an synaptic dysfunction. What I asked we're going to need a little bit of time. So we ran we've actually measured the delivery of the drug that's at the receptor in the brain. We build a model like that. What's your objective? We're all trying to figure out where to go.
Okay, if you can try to find your seats, please. We're going to get going momentarily.
Yeah, we spoke to about the program a couple of years
You make our way back to our seats, please. We make our way back to our seats. Thank you. We can make our way back to the seats. We're going to get going, please.
Come over here.
Okay, thank you very much. If you can get to your seats. All right, 2-minute warning, so let's get in our seats so we can get going and get to lunch. Okay. We're going to start in about a minute or so. There's been some questions on where the presentations are sitting. Back of your tags, if you scan, you'll get to the innovation website. And if you go down on the bottom, there's something nail, I think it's thumbnail. You'll get to all the company information, and then within that company information, there is the presentation deck that they're presenting today. So just, if you have any questions, you can come see one of us after the next slide show. Give us a second, and we'll be off and running.
Yeah, I no longer have access to the PowerPoint. I think it like logged us out. And I think we got logged out of the system. I work it out.
I know. That's what I like to think.
This isn't for
They're actually great here.
Sapan, where is Sapan? He was here?
Yep.
Yep. Can you come on?
Yep.
I think it's only 5 minutes. Yeah, it's just amazing how many. So I know, that's my 5 minutes.
And then the play button with
Okay, we're ready to go. Sorry, we had a little bit of technical difficulty. I want to shout out Luke and Emmanuel. Volunteers are doing a great job. We haven't had many issues, but let's give them a round of applause. Okay, here we go.
Okay. I'm Erik Gunther, CEO of ProteaWise. I had the same effect on the IT last time. ProteaWise was founded to solve a problem, the problem that proteomics is difficult to do at the lab bench. We all know there's been a sea change in DNA technology. It's led to little information per sample, transformed to an avalanche of information per sample. That kind of multiplexed output's led to a revolution. In genomics, unfortunately, the same thing can't be said for proteomics. 40 years ago, if you were a scientist with a sample and an antibody, you would do something called a Western blot. And basically, it's the same deal today. Western blots involve a lot of arcane equipment, a slow, cumbersome, sloppy, expensive process, and at the end you get 1 or 2 data points, and if you want to get more, you have to do it all over again. Last 10 years of my life, I've probably spent 10% of my time doing Western blots. I hate them. And everybody hates them. So my colleague Misha Kostelev and I challenged each other to come up with an alternative for Western blots. We each had our ideas. His was better, so that's the one that we're pursuing. And we call it 3D MPC for three-dimensional nanoscale protein capture. Basically what it is a novel material of our invention that we substitute for a membrane in Western blots that weakly binds proteins. Instead, we have a covalent capture of proteins. This kind of strong bond allows the membrane to be stripped of antibodies after each step of information extraction and probed with another antibody. This transforms Western blots from one and done to unlimited multiplex investigation. It also enables acceleration of the process from an overnight process to 15 minutes for each step, and it's miniaturized so that it reduces the amount of the most expensive reagent used in the process, that being antibody. So we closed our Series A few years ago. Importantly, it involves cell signaling technology as a strategic investor. They're also our strategic partner, and we're developing a new line of a new product line of single-use antibodies for use with our instrument. Our instrument will be launched in concert with this, and we'll ride their goodwill coattails for market penetration. So we've done basically 2 things since closing. We have created a cartridge that unifies all the processes involved in the Western blot. I have one here. Yep. This is it. It automates and everything from the gel to transfer to the immunocycling. The second thing that we have built is the instrument to process this cartridge in an automated fashion. So users will be able to load the cartridge with sample, insert the cartridge into the instrument, insert as many antibodies as they care to use in this carousel on the lower left, and press go and come back to quantitated, multiplexed proteomic data. This is our data that comes from this instrument. On the left, for reference, is the standard Western blot, and on the right is an overlay of sequential probings of a membrane showing the intensity of basically an unlimited number of proteins. We've done hundreds of repetitions. There's no loss in signal. Again, this happens every 15 minutes. Biologists in the audience can probably appreciate the empowerment having this kind of unlimited rapid multiplex capability would give their research. It just keeps going. And this kind of data, this kind of multiplex data is previously unknown from this kind of technology from Western blots, and it allows much more sophisticated types of data analysis and sample representation, exemplified here on the right by this heat map. It's really this combination of high information content, high throughput, low barriers to use that has driven the genomics revolution. It's what's needed for proteomics, and it's what sets our company apart from competitive multiplex proteomic technologies, as well as just Western blot. To validate our understanding of the market, we enlisted DeciBio, a well-regarded biotech consulting company, to help with a voice of the customer study. And when the technology is explained to VOCs across industry and academia, there's pretty much uniformly a high excitement, and importantly, a high likelihood that the instrument will be purchased by them for their lab at a volunteered price expectation that's favorable to our profit margin. Also at DeciBio, we assess the size of the market. Is that it? Okay. It's about $1.7 billion. Based on that information and other research, we expect to have about 700 units in the market by 2028. They'll all be chewing through our cartridges and expensive salt water that we sell to go with them, and we'll be cash accretive by that time. We're now in active discussions, and we're basically reached the paperwork stage with Thermo Fisher to act as a distributor for our system at launch. We are now raising a bridge round to get us past first sale. We expect to launch our system in end of the first quarter next year, and the bridge funding will get us to the end of that. We're about 80% subscribed. If you're interested, let me know. That's it.
Okay, thank you, Erik. And we have CCM Bioscience.
Great, okay, thank you. Well, good morning, everybody. I'd like to tell you about our company, Patent Therapeutics, and what we... Okay, thank you. So what we work on is tissue inflammation and organ fibrosis. So all types of tissue stress, as you can see at the top left, results in inflammation, and that, if it occurs chronically, results in organ fibrosis. And the area that we work in is actually liver fibrosis and liver inflammation. The two big conditions that occur in the liver are due to overnutrition, which is fatty liver disease, and secondly, overconsumption of alcohol. Both of these result in inflammation and then fibrosis. And on the right-hand side at the bottom, you can see that the injury actually results in release of DNA within the organ, which worsens the inflammation. And that's what we target. We target the receptors that detect DNA, and we block the inflammation that's caused by release of DNA. The market opportunity is very large. It's a broad perspective, but it's certainly in the tens of billions of dollars. The unmet need for both NASH and alcoholic liver disease is also very large. There's a very active drug development space in this area, and there's also an opportunity for combination therapy. Our products are differentiated because of we have human safety data for our oligo compound, which blocks TLR9, and which gives us an expedited path to IND. And specifically, this treats the inflammation component, which is relatively under-targeted in the NASH space. And in the bottom right-hand side, you can see that the two areas that we're targeting are alcoholic liver disease and NASH-induced fibrosis. This gives a summary of the individual agents. The ON TLR9 is the oligonucleotide TLR9, and then the small molecule TLR9 is our second product. The lead product, the oligonucleotide TLR9, has a clear path to phase 2 in alcoholic hepatitis because we already have safety data, phase 1 and phase 2 human safety data from a separate clinical trial. And then also we have a strong IP position with issued patents and composition and treatment claims. Here's one data set for the oligonucleotide, and what this basically shows is that in a high-fat diet model of NASH, and the histology you're seeing is that of the liver, and you can see these livers are very steatotic. That's the white area of lipid inside the liver. And after 26 weeks, which is the left image, if the animals are given the oligonucleotide TLR9 antagonist, then by 33 weeks, if they're given the antagonist, there's remarkable improvement in the steatosis, also in the inflammation and fibrosis. The next slide just shows similar data for the small molecule, which basically replicates what the oligonucleotide has shown. So development timeline would be that actually this year we're ready, if we look on the top left-hand side, for the indication of acute alcoholic hepatitis. We're ready to submit IND this year because these clinical trials are 4-week clinical trials because it's an acute inflammatory condition. And also these are inpatients, so this is a very well-controlled inpatient setting where these drugs can be given, and there's no effective therapy at the moment. So these are critically ill people which will have a 4-week clinical trial. And since there's already 8-week safety data, that IND can be filed without any additional work that needs to be done. And then the phase 2 trial for acute alcoholic hepatitis could actually start early next year and be completed by the middle of 2025, with then subsequent development after that. For the clinical trial for NASH, which would be at least a 24-week or perhaps a 52-week trial, we would need to conduct some chronic toxicity, but then the IND filing could be by early 2025. What we are seeking is pre-seed currently and to initiate the clinical trial for acute alcoholic hepatitis, which would be for $2 million. And that, as I said, with the IND filing would be this year. And then the first tranche would actually complete the phase 2 for the ALCAP and conduct the safety study to allow for IND filing for the phase 1 for the NASH study, and then the second tranche for $25 million would then complete the phase 3 study for alcoholic hepatitis. And I'll stop here. Thank you.
Okay, great. So we got Dan Carlson, Saraxion.
Timer's there.
All right. All right. Hello, everybody. My name is Dan Carlson, and I'm the CSO of a large animal genome editing company. And we've had the pleasure of working with Yale for the last several years to develop Sarsion biologics. And our mission is to create a brighter future for patients with the world's first gene-edited biologics that enhance the body's natural healing ability. So Sarsion is a spin-out of Recombinetics bioincubator model. So we have core competencies and shared services that really boost market-driven opportunities in this space. So we obviously focus on the gene editing and the animal development at Recombinetics, and we've boosted companies such as Excelgen, our large animal ag company, and Makana Therapeutics, which is a xenotransplantation company. What we're here to talk about
a big problem, and that's chronic wounds. So due to aging populations, obesity, and diabetes, we're seeing up to 2% of people worldwide that are developing chronic wounds. In the diabetic space, in the United States alone, we're spending over $100 billion treating these diabetic wounds. And what the patients go through is excruciating and it's heartbreaking, and the life expectancy of a patient that develops a foot ulcer is actually worse than many cancers. So what are the roadblocks in chronic wound healing? So we first look at poor cell migration. You have to get the right cells at the wound site to mediate the repair that's required. Secondly, we have poor blood supply, so lack of oxygen reaching the site, obviously the cells, and the nutrients required for healing. Finally, we have poor tissue integration and remodeling at that site in a lot of these chronic patients. And even in the use of some of the advanced therapies out there, we get poor integration with the substrate, leading to poor healing. So what are some of these substrates? So we're talking about extracellular matrix, which is an essential component of the wound healing process. And essentially what that is, it's the substructure of all of our organs and skin and tissues. And it's interconnecting protein substructures. It enables cell attachment, migration, growth. And it's been animal sourced for over 20 years and used in the wound treatment space by some very big multinational companies that you're seeing here. But we found that not all extracellular matrix is created equally. So this work began with Themis Kyriakides here, who's in the room. He worked on some mice almost 20 years ago, or 25 years ago almost, and found that they had an interesting wound healing advantage. Doing a lot of follow-up studies, still to date, has found that the extracellular matrix enhances patency of vessels. It's got an altered structure. But the main finding that's important for us here today is that it's not a cell intrinsic property. It's a property of the extracellular matrix that's secreted by the cells, which enables us to extract that in any cellular way and produce products that can help patients. And we teamed up, developed a gene-edited pig that has the same modification of TSP2 that we can generate these products from. Long story short, the extracellular matrix from these animals is able to enhance cell migration. It improves the blood supply and angiogenesis, and it improves the tissue integration and remodeling. So our lead product is dermal hydrogel, Sarsion DH, which can enhance wound healing in diabetic patients. It's a 510K eligible product, so this is not an IND or a BLA that requires 7 to 10 years. This would be a 6-month timeline from submission. And it can be a solubilized ECM using well-established protocols already, and it can be injected or molded. Right here you're seeing a demonstration of the Sarsion DH in action. The blue cells here are integrated well into the tissue on the Sarsion DH side, where wild-type hydrogels that are the normal products out there, we see poor migration and lack of integration there. Also importantly, when tested in diabetic mice, we found wound healing to be about 2-fold enhanced. When we take that product from a mouse setting and generate it from a pig, we can replicate those results. And we can see here on a wound that was created and treated with different agents, the Sarsion gel on the far right, we see some very robust epithelial layers forming on top of the wound. We also see very mature collagen, indicated by the bright blue there. In the wild-type versions or the untreated animals, you're not actually seeing any epithelialization, which is an important factor in healing. So we've mapped out the lead product pathway, both regulatory and development side. We've been able to identify a very great company, Collagen Solutions, right in our backyard, that is a key manufacturer for a lot of these projects for multinational companies. We're going to be piloting large animal studies soon and do 510K and commercialization. And we estimate we'll need about $5 million to fund this and continue to drive our follow-on products. And what those products are, so we are able to use everything in the pig. And we found that we have enhanced healing from not only dermal hydrogels for wounds, but also in bone, muscle, and then other tissues that we can utilize from the animal that also have some preclinical efficacy demonstrated. And we think this would poise us very well to be in a launch position, exit position shortly thereafter. And this is just the introduction of the team. Obviously, Themis Kyriakides here on top, and a bunch of veterans in the wound healing space, and both in regulatory and commercialization. So thank you for your attention.
All right, great. Sapan Shah from Stratify Bioscience is next. Okay, so we need a minute. So, reminder that all the slides are on the innovation website. You can get to that website by scanning the back of your badge and getting down to the file thumb or something of that nature.
Thumb raise.
And you'll find company profiles as well as the presentations within those company profiles. Obviously, people are here after the presentations as well. We have a couple more to go.
No video.
Perfect. Okay. Thank you. I was wondering how I was going to finish my presentation in 5 minutes and then learned that I had no slides, would solve that problem, but maybe not so interesting for the audience. But thank you. My name is Sapan Shah, CEO of Stratify Biosciences. We're an early stage EL spin-out, pioneering cell adhesive nanoparticles to enable innovative site-directed therapeutics. So the challenge we're trying to address with our platform is targeted therapeutic delivery. And simply put, the goal is to deliver therapeutic payloads to diseased tissues while avoiding off-target absorption, which leads to negative outcomes for patients. There's been a lot of innovation in the nanoparticle space. Most of that has been focused on systemic delivery, and systemic particles tend to accumulate in the liver and don't get to their desired targets, leading to poor therapeutic margins. Our solution to this issue is bioadhesive nanoparticles, which we call BNP. And these are particles that have been engineered for cell adhesion, uptake, and payload release. And we can load a wide range of payloads, encapsulating them in the polymer, ranging from small molecules, cytotoxics, peptide nucleic acids, and nucleic acid oligomers. The platform for the company was licensed from Yale. We've been seed funded by Elm Street Ventures. So made some great progress. We have in vivo proof of concept with a lead program we're calling Stratify 101 for multiple oncology applications, and I'll share a snapshot of that data in a second. And we're seeking funding now to advance the lead program into the clinic in 2024, and are actively in discussions with partnerships to expand the pipeline to different therapeutic applications. We have a great team on the ground. The company was founded by Marc Saltzman and Michael Girardi, who have been fantastic to work with. I jumped on board as CEO a few months ago. I've had the fortunate opportunity to lead about a half a dozen biotech companies over the last 20 years, and working with Brian Dixon, a venture partner at Elm Street Ventures, who's built the company up to where it is today. A little bit on the particle. So what we are working on, which is developed in Marc Saltzman's lab, is a polylactic acid polymer core. PLA is a material that's been used in a lot of medical applications in sutures, medical devices. In this case, we're generating nanoparticles that are 80 to 100 nanometers in size and using that to encapsulate a drug payload. The real innovation here is in the coating. It's an aldehyde coating that binds to proteins that are proximal to the site of administration, including proteins on the cell surface. So that allows cells to retain and uptake the particles and release their payloads in the cytoplasm and most importantly to the nucleus for different payloads. We get payload release over days and weeks, limiting systemic exposure, which is a really nice profile. And we think this opens up a new paradigm of treatment. With platforms like this, the challenge is always, what's your first killer application? What are you going to focus it on from a target perspective? So we've chosen to go after a couple oncology targets by encapsulating a really potent cytotoxic agent inside the particle. We've generated some really interesting data both in non-melanoma skin cancer and ovarian cancer, which I'll show in a second. So quick snapshot of a mouse model, proof of concept mouse model of a squamous cell carcinoma. The key takeaways here are on the left, we get retention of the particle out to 3 days and beyond versus the free drug, which really it does not hang around much longer than a day. That translates to a 70% reduction in tumor size up to 14 days and a really dramatic improvement in survival in these mice. Second application is intraperitoneal treatment of ovarian cancer. The key takeaways here on the left is an intraperitoneal administered BMP particle with the cytotoxic agent retains in the peritoneal lining for up to 2 weeks and beyond. We see a little evidence after 21 days still of the particles hanging around. That translates into really effective elimination of the ovarian cancer cells. On that middle panel, you see after 49 days, almost complete elimination of the ovarian cancer cells versus the free drug, which is not as effective as reducing the cancer. And similarly on the right-hand side, you see just a dramatic reduction in elimination of cancer cells in this application. So we think with this proof of concept data, we have a couple of really interesting targets to pursue. Non-melanoma skin cancer, it's a huge issue. There's 5.5 million new cases each year in the U.S. alone. Standard of care is surgical excision, but that's really not feasible for a lot of patients, particularly depending on the location of the tumor and whether you need multiple surgeries or reconstruction. So we're excited about this application. With peritoneal cancers, 75,000 new cases each year, and we think there's a huge opportunity to improve upon standard of care here, which is cytoreductive surgery plus HIPEC. So we're excited about these two. We're in the process now of raising funds to advance the platform. We have a lead program focused on those oncology targets. We have a pharma partnership focused on an immunology target, and looking to advance these into the clinic over the next few years. And these are some of our key milestones coming up. And be happy to chat with anybody who wants to learn more. Thank you.
Okay, we have Translura, Stephen Wang. Thank you. Timer is in front. All right.
All right, so my name is Stephen Wang. I'm an assistant professor of genetics and cell biology here at the Yale School of Medicine, also co-founder of Translura. Our company is a spatial omics company that offers a new tool to map the new dark matter of the genomes. Our genome is folded in a 3 dimension in a cell nucleus in a hierarchical manner across many length scales. And these different 3D genome architectures, they control many essential genomic functions and show intriguing dynamics and changes during development, aging, and many diseases. But our understanding of the 3-dimensional genome folding is in fact still much less than our understanding of the 1D genome sequence. And the 3D genome is considered the new dark matter and a new frontier in genomics. Mostly, we don't know largely what is the 3D folding path of DNA at length scale above the nucleosomes. And the reason why we don't know this answer for decades is because technical limitations. So previous techniques yield DNA blobs rather than the really 3D folding traces of DNA. And they are usually not directly in 3D, and they often require population averaging. So not really good for a context of diverse cell types and states in a complex tissue, such as in tumor. So to address these limitations, several years ago, I invented a first-in-kind image-based 3D genomics technology called chromatin tracing that sequentially image, pinpoint, and distinguish numerous genomic regions along individual chromosomes in single cells, and really enabling this direct tracing of the 3D folding organization of DNA in situ. So this technique opened up this field called image-based 3D genomics and have very broad applications in research, diagnostics, and drug target discovery. For example, in our most recent work, we applied this genome-wide chromatin tracing in a mouse lung cancer model, generating the first ever single-cell 3D genome atlas in any cancer. And by applying machine learning to the new 3D genome data, we were able to demonstrate new diagnostic strategy of cancer states. We were able to discover novel cancer progression driver and suppressor genes in lung cancer. So these are new cancer therapeutic targets. And of course, we were able to generate a list of new insights in basic cancer biology as well. And in another work, we further combined our chromatin tracing technology with the CRISPR technology to generate the first ultra-high content, image-based CRISPR screening platform. And this platform allows us to systematically discover new regulators of the 3D genome and the spatial transcriptome. And our vision here, these new spatial omic regulators are a new class of therapeutic targets and genome engineering tools. So our company, Translura, currently is offering this chromatin tracing technology in an end-to-end fashion. Our offering includes probe library design and construction, tissue sample preparation protocol, automated machinery to do this work, and data analysis pipeline. We have made a first sale to the Yale Center for Genome Analysis back in mid-2021. So our current business model is instrument sales plus reagent plus service contract for the instrument. But as shown before, we have actually potential to expand to other modes of business model as well. The spatial market field is really entering a rapid phase of expansion with multiple independent studies indicating the $500 million market size in just a few years. And within this rising field, our company, Translura, has its unique competitiveness and strengths. So most companies in this rising field focuses on spatial transcriptomics and proteomics, as we heard from the talk before, and only 2 companies focuses on directly mapping the 3D genome folding. And in comparison to the other company, our company, Translura, is clearly more advantageous in terms of genomic throughput, the application in complex tissue, multi-omic capacity, and also demonstrated the product sales. We also have exclusively licensed multiple technologies from Yale. We are a very strong team with strong members on both the academic and the business side. We have also received the $150K convertible note seed money from Connecticut Innovation. And we are now trying to raise $3 million round pre-A or A round to expand our operation and achieve our next milestones. So in sum, we are Translura. We are mapping the new dark matter of the genomes. Thank you.
Okay, we have Reed Waldman from Vrselja.
Thank you very much.
Timer there.
And I take it this is advanced. Yes, big button. Well, hi everyone. I'm Dr. Reed Waldman, CEO of Vrselja, and I'm really excited to tell you how we're transforming medical dermatology. Now, I co-founded Vrselja in 2019 with another board-certified dermatologist around the idea that there are conditions that we see on an almost daily basis in our clinical practice for which there's been no meaningful innovation in decades and for which there's really no meaningful innovation on the horizon. And so we set out with the mindset that we could take our end-user knowledge as dermatologists to really transform some of these conditions. And since then, we built a business that is centered around 3 pillars within dermatology. One is that we focus on developing within under-innovated markets, or markets where big pharma has not spent substantial capital. 2 is that we're a portfolio company with a diverse pipeline of 5 first-in-class assets that I'll tell you more about. And the 3rd is that we target lean capital efficiency to enable that portfolio model and augment returns. And using this strategy, we've accomplished quite a bit over the last 3 years. We advanced our lead product into a phase 2 study. We have another product that we are initiating a phase 1 program for, and the remainder of our pipeline is poised to enter the clinic within the next 12 months. We've also shown that we're committed to a lean development approach. With less than $5 million of capital expenditure, we were able to get that lead program into our phase 2, and we're working right now on building robust BD infrastructure for inbound, outbound BD, and we're well capitalized right now with more than $25 million cash on hand as of our last monthly close. As mentioned, we have a portfolio of 5 products. These hit indications including warts, androgenetic alopecia, molluscum contagiosum, alopecia areata, and atopic dermatitis. And as mentioned, over the next year, we believe that these programs are poised to enter the clinic in a variety of phases. Given the time limitations, I'm going to focus on telling you about our wart program. And this is really the program we founded the company around. Common warts, as their name suggests, are incredibly common. In fact, 19 million Americans are affected, and 2.2 million Americans go to the doctor for treatment of their warts every year. And despite being common, there are no FDA-approved treatments for common warts, and the treatments that are used, like freezing with liquid nitrogen, are either painful, they scare children who are disproportionately affected by the condition, or they require daily applications for months at a time, which is simply too burdensome. And my co-founder Tim and I were familiar with a treatment paradigm called intralesional immunotherapy, where a foreign protein is injected into the wart to activate the body's own immune system to recognize and fight off the wart virus. And while this is well established, known to work in roughly 70% of cases, it has really two limitations. One is that it's injected, and we're talking about injecting primarily into digits, which is exquisitely painful, consistently one of the most painful things I did in my clinical practice. The second is that it has a lot of logistical issues related to its approval landscape. And so we set out to democratize that treatment by incorporating it into a dissolvable microarray, where it's applied like a Band-Aid, process that's virtually painless, left on for really like 5 minutes, applied to a single wart to treat all of a person's warts, and then thrown out. As mentioned, this is in a phase 2 study that we hope to be phase 3 enabling. We have a readout for this study coming in the fourth quarter, and based on independent commercial assessment, given the high prevalence of this condition, the top 50 most prevalent condition that affects humans, we anticipate substantial revenue potential. As mentioned, phase 2 is ongoing. Wish I could tell you more about it if we had more time, but we intend for this to be a phase 3 enabling phase 2 study. We are very confident in the readout that we have later this year because we have great data for both the microarray and for the biologic product that's in the microarray. We believe that this product is going to be a long-term revenue source for the company because it has very strong IP position, including issued patents, as well as a great regulatory environment. It's a de novo biologic product, potentially with 12 and a half years of regulatory exclusivity. In short, it's a product that, again, targets a top 50 most prevalent condition in humans for which we have low scientific risk based on known efficacy of the biologic product. We have a limited competitive landscape with no FDA-approved treatments, limited treatments in the pipeline, and favorable landscape that allows for long-term revenue stream, cornerstone potential for this product. We have a wonderful team, wonderful investors, one of which I hope everyone gets to see in a panel discussion later today. And we're very excited to have introductory meetings, not raising capital currently, but would love to get to know you all, tell you a little more about the remainder of our portfolio. Thank you.
Fantastic. We have 3 more. We have Demetrius Braddock from Intellion.
Okay, great, guys. Thank you very much. A lot of exciting science. I'm here to tell you about a new project coming out of my lab. I'm at Yale. My lab turns enzymes into drugs. Previously, we developed the lead therapeutic for Inozyme, which was shown to be potent, safe, and effective in phase 2 data released in February of this year. It's moving into babies, actually, with this lethal disease, and phase 3 will begin this year. The current project is focused on this problem, which I can't point to, which is extracellular DNA in the blood. This either fights infection, but if it's not cleared, it leads to autoimmune disease. So this is not a new idea. It's been out for 70 years. So for 70 years, the first patients were identified with loss of tolerance of self-DNA in their blood with lupus. 40 years ago, we found that patients with enzymes that degrade this DNA had low activity in these patients, and 20 years ago, actual loss of function mutations in mice and in humans with autoimmune disease were identified. So this idea has been around for a while, but it's fallen into oblivion because in 1999, attempts to treat lupus patients with the recombinant enzyme to degrade DNA failed, and they failed, if you read the article, because that enzyme was not bioavailable. But beside the point that it wasn't bioavailable, it basically lost steam, and that's where we are today, 20 years later. The march of science progresses slowly. So what we've done is we've developed an enzyme now to do this. It's on a neutral metalloenzyme scaffold. It degrades both free and chromatin-bound DNA, so it has the activity of both DNase-1 and DNase-1L3. We produce it in CHO cells that are supplemented with enzymes to help the glycosylations. We purify it in GMP resins and techniques. When we inject it into a mouse at 1 mg per kg, once a week we have full activity for an entire week of the ability to degrade both free and chromatin-bound DNA. So that took us 3 years to do. And what we're going to do now is use it in various models of autoimmune, autoinflammatory disease. I only can tell you about one. It's called diffuse alveolar hemorrhage. You might not have heard about that, but you've heard about COVID-19. This is how COVID-19 patients die. They basically react to the virus, and they develop DNA in their blood, and they get a cytokine storm, which triggers the DNA damage response, interferons, and they bleed into their lungs and die. There's no specific treatment, just supportive care, ECMO, mechanical ventilation. You've heard it all before. This also occurs in sepsis, it occurs in lupus, it occurs in encapsulitis, and a lot of other diseases. So it's a nonspecific autoinflammatory response due to free DNA in your blood. The global sepsis market's quite high at $430 billion. So what we're going to do is we're going to treat the mouse model with this. In mice, what we do is we stimulate them with an inflammatory agent. It causes cell apoptosis, releases DNA into the blood. We do that on day 0. We wait for the first mouse to die, and then we begin to treat with this enzyme subcutaneously. And you can see in this Taconic Black 6 strain that 70% of the mice die without treatment versus 30% that are treated. In another strain of mouse, Jackson lab mice, we have 95% survival with treatment versus 70%. So it depends on the genetic background of the mice, how they respond. Just like in the human, the mortality is from 25 to 80% mortality in humans. The mice have diffuse alveolar damage by gross and microscopic histology, and they also go into a coagulopathy, which occurs in humans as well. So humans will bleed out because they develop DIC. These mice also elevate D-dimers, drop their weight, and die in a similar mechanism. So that's my story. We are about 27 months away from first patient in. With the proper funding, we have a biologic, doesn't need further modification or optimization. It's ready to manufacture. We're looking for partners to help us do this, to get this into patients for a series of autoimmune and autoinflammatory diseases. I'd love to talk to anyone interested in this further. Thank you.
Okay, we're in final stretch here. Paul Turner from Felix Biotechnology. Paul, nice.
Yeah. Okay, thanks for hanging in there. You're almost to the break. I'm very happy to be representing Felix Biotechnology, a company that was formed using discoveries right down the street in my laboratory here at Yale University. So I'll give a high-level overview of some of our new technology that we're developing. So we're living through a pandemic, and that is very difficult, but it is also a reminder that viruses have amazing potential. They also have amazing potential to solve problems, and that's what we focus on. So we're building a virus design platform that is nimble and broad because we want to take machine learning and AI tools to design custom phages. These are viruses that kill bacteria, and we want to do this in order to, in essence, engineer any phage for any problem that bacteria cause. And bacteria cause lots and lots of problems. It has a huge market opportunity in the fact that the role of bacteria in health and disease, microbiome health, women's health, personal care, and skin care is quite obvious. Also, bacteria threaten the lives of our animals and plants and therefore food security. They also contaminate our water. And they'll also interfere with our manufacturing and lead to things like methane emissions that we care about for global warming. So there's a lot of opportunity to control bacteria, but the problem is antibiotics, chemical antibiotics don't do that very well anymore. So instead we want to deploy phages. These are safe and effective biological drugs, and they are alternatives to chemicals. So they kill bacteria, and they do this with high degree of specificity, which is quite different from something like a broad-spectrum antibiotic. They have low toxicity, and they often have a novel mode of action compared to other chemical antibiotics that we've relied on historically. You can administer them in low doses because they can make copies of themselves. They can also target biofilms, which are really the structures that make bacteria impermeable to chemicals that might kill them. And indeed, all of this is chemical-free. So if we seek solutions for tomorrow that are in the absence of chemicals that might contaminate our environments, this is really a way to go. So one thing to focus in on is the microbiome. We know its role in health, and it is desirable to take something like a phage and prune something out of the microbiome in order to control an invader that has come in as a pathogen, or in order to reset dysbiosis and make the microbiome more healthy. So we have lots of applications for phages as microbiome editors. They can remove microbes that drive disease, and they can validate the role of some strain or species in disease in terms of a diagnostic. So there's a huge potential for phages to restore microbiome balance as we learn more and more about the role of the microbiome in health. In humans, plants, and animals, and other creatures, this is really the way to go to engineer microbiomes using phages. So the problem is that there are a lot of ambitions to use phages, but it's not easy to do this. And the reason is that they tend to be hard to engineer. They actually represent lots of different biology, and we need to have a common way to easily engineer them for different purposes. So that's what we're developing at Felix. It basically allows any phage to be simply engineered. They often have narrow host ranges, which is not so desirable for covering so-called genotype space, but we can expand their host ranges very easily with our platforms. And also, as an evolutionary biologist, I'll tell you that my concern is that you deploy a phage, it selects for resistance in the bacteria, and it fails. So therefore, we've developed technology that will mitigate this problem. So our platform could be summarized as design, build, and test. In other words, we bring rigor. So we want to be able to use machine learning to inform the design of viruses to solve any bacterial problem. We want to be able to do this easily in a test tube with a print and boot approach, where we use Golden Gate assembly to essentially create any virus for any purpose. And we have high throughput tools to make sure that we do this accurately and safely. So we have a growing IP portfolio. That's the focus of today, is to emphasize that we have high throughput characterization to be able to find phages to solve problems. We modify them further through machine learning if necessary. We use proprietary approaches, CRISPR-Cas13, with technology to nimbly and genetically engineer any phage that you want. And essentially we are using synthetic phage biology of tomorrow, and we're doing it today. We have this broad IP. I don't have time today to really emphasize how this is safe and effective. We've used our phages in patients at Yale New Haven Hospital and elsewhere in over 50 personalized treatment, very safe and effective approaches to, say, clearing a bacterial problem that antibiotics will not. We just finished a phase 1/2 clinical trial on this. So we have a lot of ambitions for the future. We want to be the one-stop shop for people who have bacterial problems as targets, and we'll be able to engineer phages to solve them. Thanks for your attention.
Okay. Last but certainly not least, Chris Duke, Genoa.
Yeah, probably.
Hi, good afternoon, everyone. I'm Chris Duke. I'm the CEO of Geneo Bio. This work comes out of the lab of Dr. Peter Glaser here at Yale, and we're very grateful for his continued involvement, along with one of our other scientific co-founders, Elias Chiano, who's here today as well. I'm going to talk to you about our GMAB platform technology. This is a delivery platform, a unique delivery platform that takes advantage of a very unique antibody that has been around for quite some time. And it very much complements a lot of the work that has been done in gene therapy over the years, while at the same time fundamentally addressing some of the unmet needs that continue to exist within gene therapy. So the best way to understand this approach is to really break down the fundamental components of this approach. At the center of all this is this unique antibody, and it's been around for quite some time. It was identified back in the 90s as an autoantibody that has the ability to penetrate into cells and to bind to DNA. Dr. Glaser and his team continued to do some additional work on this and specifically identified the binding pocket on this antibody where it binds to nucleic acids, and it happens to be in the CDR region. Further work was done by Dr. Glaser's lab where he showed that not only does this antibody bind to DNA, it has the ability to bind relatively agnostically to a wide range of nucleic acid payloads. So this property fundamentally elevated this to the status of a broad delivery platform. And with the continued work that we've done on this, in addition to nucleic acid payloads, there's also additional flexibility that allows for binding and delivery of a wide range of payload types, including cytotoxic payloads, CRISPR-Cas9 payloads, and small molecules. And the third piece that rounds out this story is the targeting mechanism for this platform. So this antibody and these antibody nucleic acid complexes interact with and are taken up through a unique nucleoside transporter called ENT2. And while ENT2 is expressed on most cell types, it's very highly differentially expressed, specifically on tumor cells and skeletal muscle cells. Essentially, cells that tend to either have high metabolic activity or are highly proliferative are going to have highly elevated levels of ENT2. So some data here on the right, specifically, this comes out of the Cancer Genome Atlas, and what you can see is that ENT2 levels not only are consistently highly expressed across a broad range of tumor types, but they're also highly expressed consistently within specific tumor types. And we estimate that there's about a 1 to 2 log-fold difference in expression between tumor cells and non-tumor cells. On the left, you see data in non-tumor tissue, and you can see clearly that skeletal muscle has a very high abundance of ENT2 expression, and it's about a 4 to 10x fold difference compared to other healthy tissues. And this is really the key differentiating factor, probably the most important slide that we often present. And it has to do with the unique mechanism by which these complexes internalize into cells. So this antibody does not rely on a protein-protein interaction with some type of cell surface antigen that gets taken up through a traditional endosomal lysosomal pathway. And while we don't know the specific mechanism, we do know that these complexes have the ability to internalize into cells and pass directly into the cytoplasm of the cell and ultimately into the nucleus. And what you can see here is that we did an experiment where we used 2 different antibodies. First antibody labeled against GTP-RAB5, which is a marker for early endosomes, and a secondary antibody against our GMAB antibody. And the images you see on the right here after incubation were taken about 1 hour after incubation. And what you would expect to see if this was co-localization into the endosomes is that there would essentially be an overlay between the pink and the purple in that bottom right panel. But you can see clearly that there's differentiation around the nucleus of the antibody. And we've further gone on to show that this approach has ruled out other endocytic pathways. In the interest of time, I'll just move through this quickly. This is actually auto-generating here on the left. This is some live cell imaging. And what you see is that within 24 hours, 85% of the cells are able to take up this GMAB antibody. So there is rapid uptake into the nucleus of cells. And we've gone on to conduct this experiment not only across